Hey everyone, welcome to this new episode of the Intelligence Podcast. This time I had the pleasure of speaking with Dr. Brian Kennedy, a director of the Center of Healthy Longevity at the National University of Singapore and the former president and CEO of the Buck Institute for Research on Aging. Dr. Kennedy is one of the pioneers in the field. I had the opportunity of learning more about his perspective on the progress in longevity research and also about his personal contributions to the field. I mainly focused on Dr. Kennedy's opinion and took his advice about the use of drugs and supplements that are usually claimed to improve health and lifespan. You can think of rapamycin, metformin, AKG, for example. I also focused on Dr. Kennedy's own contributions on research on drugs and supplements. And as usual, I discussed the role of AI and its importance in accelerating research in that field. And finally, we discussed the social impact of humans potentially living a longer life. That was a really interesting discussion, and I'm glad you're joining me here. I hope you enjoy. Okay, so let's start by talking a little bit about perspectives. So I actually see many research in the longevity sphere. I also see, as everyone who is following the domain a little bit, many companies actually created in that sphere, many funding behind those efforts. So this makes me question what expectations actually we should wait or look at for that field. Like, what are your expectations in terms of the time frame within which we are going to start to perceive impact on our lifespan?
Well, actually, I think the time frame is yesterday. And I'll explain what I mean by that. Because some of the most effective drugs that have been used to treat disease, and after all, what we've been really doing in healthcare is not healthcare, but sick care. It's trying to identify your risk of disease, treat your chronic diseases. But many of the most effective treatments are very early stage. They're actually preventative. So metformin for high glucose, statins for high lipids, fibrates, other drugs, beta blockers. These are things that reduce the risk of early stage disease progression, and they've been very effective at allowing people to live longer, even though they're heading down some path toward disease. And I think there are things like metformin are so early stage intervention that they're at the interface between aging and disease. Aging is the biggest risk factor for diabetes and everything else you're worried about. And there's kind of an interface, a transition between your age and your going into a disease state. And I think these drugs are acting at a very early stage, and they're actually targeting aging pathways as well as disease pathways. And that may be why things like metformin are candidates longevity molecules as well, because the interface between metabolism, aging, and diabetes is so tight. So, I'm not sure that's exactly what you're asking, but I think that we're already starting to treat aging. We just didn't know about it. Um,
This is actually my question. It's, let's say, surprisingly optimistic answer, actually. So, what you mean is that some people who are already taking those drugs might, let's say, witness some extension to lifespan, like within a few years.
I think it's possible, yes. I mean, we thought we were treating disease when we developed those drugs, but actually we may have been treating aging as much as disease. Yeah.
Okay, so if we will talk about supplements a few minutes from now. But I'm also interested because metformin is an existing drug, like it's been
Yeah.
years, it's in use. So, do you have how many years, maybe more or less?
I don't know, but decades. I mean, millions of
Yeah,
people have taken metformin. The interesting thing about it is we know effectively what it does. It reduces hyperglycemia. It improves peripheral glucose uptake for the blood. Um, but we still argue about what the mechanism of action of the
yeah,
drug is. And a lot of these early stage drugs that were developed, they weren't defined based on the mechanism. They were just found based on other properties. And so we still are not quite sure exactly what metformin's doing. And I think that's true of a number of of drugs that were developed in the early stages. But we know that it's improving metabolism, and I think there's no doubt that it helps people that are pre-diabetic, that have high glucose. The question is whether it's going to be beneficial for people that are not in some stage of metabolic disease, that have normal glucose metabolism. Will it still affect their aging? And I think that's the question that we need to answer. And people like Nir Barzilai are trying really hard to get clinical studies done on that.
Interesting. Okay, so we have these popular 9 hallmarks published by Lopez et al. In 2012, which also followed the sense strategies proposed by Aubrey Gray a few years before this. So they all seems to agree about some mechanisms. What I find interesting in the Lopez paper, is that they classify those hallmarks in, let's say, categories of hallmarks. So the first category is the primary hallmarks, or the root, like real root causes of damage. And then there's the second or secondary category, which is the mechanisms that are responses to damage. And then there's the last one, where the real mechanisms that are directly related to phenotypes, or the damage that we are seeing, let's say maybe skin or some other diseases that we perceive. So I'm going to ask you about some of those uh, main causes. So when we talk about primary hallmarks, so here we talk about genomic stability, telomere attrition, etc. So it seems to be omics related issues, things that are in our genetics, genomic code, and all the things around it. So when we talk about genomics instability, what do we refer to by saying this? Where does this come from actually?
Well, genome instability would be mutations to your DNA. They get your body's actually quite good at repairing DNA mutations, but certain types of mutations are hard to repair, or they change the genetic code when they're repaired. I mean, telomere attrition itself is a form of uh, uh, chromosomal damage, attrition of the ends of your chromosomes. And so this type of damage accumulates with age. And I think maybe more importantly, our capacity to fix that damage goes down with age. And that's certainly one of the drivers of aging. But, damage is happening to uh, all sorts of molecules in your body, proteins, lipids. And I think those contribute to aging too. Loss of proteostasis is another one of the hallmarks, or certainly was one of our pillars of aging. And lipid. I think we don't think enough about changes that are happening in lipids with age as well. So I think that damage occurs at the cellular level, and certainly DNA is one place it's been looked at a lot. And I didn't mention epigenetics, but epigenetics is basically how your genome is packed in the cell. And how it's organized, because in some cells, like muscle cells, you need to express muscle genes and make muscle proteins. Whereas in other cells, maybe they're skin cells, you need to make express skin genes and make skin proteins. So how you organize the chromosome depends on allows certain genes to get expressed and not others. But that organization seems to deteriorate, or there's certainly noise in that system as you age as well. There are epigenetic changes, and that can be driven by the environment, lifestyle, stochastic events, and a variety of other things. So, again, that's a form of loss of information. So, I think those are probably key drivers of aging. One of the things I would
Yeah.
point out, though, is that it hasn't been extremely successful so far to fix a single hallmark of aging and see that have an impact on the lifespan of the organism. So if you look at mouse studies, or invertebrate studies like worms and flies, or non-vertebrates like yeast, uh, it's not really fixing specific problems that seems to be impacting longevity. It's more signaling pathways that seem to be at nodes of these hallmarks. So I think of aging a little bit differently. Certainly, all of those things impact aging, but that doesn't mean that fixing one of them is going to have a significant impact. I think that what we've seen so far is that the interventions that extend lifespan improve many or most of the hallmarks and pillars of aging. And it may be that healthy aging is having a homeostatic control in your body. It's maintaining equilibrium, even though these things are happening. And your body's very good at doing that for a period of time. But when enough things go wrong, that sort of network breaks down. The drugs and genetic changes that really impact aging seem to be preserving the network. And the reason I say that is if you look at all of the hallmarks and pillars, they're all improved.
Yeah,
So it's not as if they're fixing one thing, it's as if they're restoring a network that maintains everything longer. And so I think that's where we are right now with interventions. Now, it may be that if we could perfectly fix DNA damage or oxidative damage or had the right mechanism to target, damage to proteins, proteostasis, we might be able to have a big impact on aging by just
yeah,
targeting one. But so far, that hasn't been the model for the interventions that have been developed to
yeah.
date. So I think of it more as a systemic process.
So this is an interesting view, but don't you think like originally the hallmarks were suggested to, let's say, avoid uh, thinking in terms of the very complex network of interactions. So we, like the community, tried to, let's say, extract some main causes in the hope that if we treat one or more of
Yeah.
them, we will be able to gain some time. So what you're saying is that even those hallmark also represent a very complex network of interaction, and fixing one of them, it's not clear that it will actually work if we don't work on all at the same time.
Yeah, I think that may be what the data is telling us. It's not
Mm.
as if we set out to do it that way. I mean, the mini labs found different drugs and genes that affect aging. And I think one of the first ideas everybody had was to say, all right, they worked backwards. They found, say, rapamycin extends lifespan. Let's figure out which hallmark it's affecting. But then it's improving a lot of them. There's less inflammation. There's less
Yeah,
damage from senescent cells. There's better proteostasis. There's better stem cell function. And so it wasn't like you could attach one intervention to one hallmark. Now, if you go the other way and say this don't find drugs randomly, this actually go and target hallmarks. So far, that hasn't been that effective. Now, maybe it will be, but I'm optimistic that there are many strategies to extend health span and lifespan. But so far, that hasn't been the most effective approach. And
okay,
even if you do target one pathway, the kinds of things you're hitting are the same interventions that have systemic effects. So you know
yeah,
so these things are just very connected to each other, and we tend to take a very reductionist approach in biology, and I think that makes sense. We want to understand the details of how things work, but if there's nothing more systemic than aging, and so it
exactly.
affects every tissue. It affects, it's affected by everything you do in life, and so I think that this may be a case where it really is a complex network that we need to understand. Having said
Yeah,
that, it's still true that single genes and single drugs can affect the process. So even though it's complex, that doesn't mean we're going to need a combination of 38 drugs to extend lifespan. You can do it with one drug or one genetic modification, at least in mice, and I think that the evidence is starting to accumulate in humans as well. The
yes.
effect size may not be huge, but at least that's where we are right now. So
Yeah, so what I understand is this strategy is even if you don't treat aging as a whole, let's like gain 1 or 2 years each time. Like if we find some really high impact drugs or strategies, let's gain some time. So here I'm referring to the second type of hallmark, let's say second and third, maybe the antagonistic or the responses to damage. For instance, cellular senescence and the nutrient sensing.
mhm.
So we suggest that if we clean out those senescent cells, for instance, we might be really having big impact on extending aging. So first, can you just like mention what is cell senescent and nutrient sensing, and why does that affect aging?
Yeah, so all of your tissues are dividing at different rates, but they're dividing. Some very slow, some have very little proliferation, like neurons in your brain. Others are like skin is proliferating all the time. Um, but so things can go wrong with this proliferation process as you age. If you get hyperproliferation, that can cause cancer. Everybody knows that cells dividing when they shouldn't divide. But it's also true that the cells exhaust their potential to divide in the body. They can do that just by dividing too many times. There's a limit to how many times cells can divide, or it could be through stress or other things that cause the cells to go into this non-proliferative state called senescence. And people have thought for a very long time that senescence may be a factor in the aging process, uh, largely originally from cell culture experiments. You took primary cells and put them in culture in a dish, and you just let them grow, and they stop growing after a while, and they develop these other properties. Um, and then people said, well, I wonder how many senescent cells are there in the human body or in a mouse when it ages. And people found a very low percentage. So a lot of tissues had no more than 5% of the cells, 5% of the cells senescent. And then that kind of made people think senescence wasn't that important for aging. But then more recently, work by Judy Campisi and other labs has shown that there's something called a senescence-associated secretory profile. So all cells secrete things into the bloodstream or into the neighboring environment that they're in. But when cells become senescent, they change the repertoire of proteins that are secreted. So they now secrete things that are very inflammatory and have adverse effects on neighboring cells or even systemically when they get into the bloodstream. So it seems like even though only a small percentage of cells may be senescent, they can actually secrete factors which cause problems for other cells in the environment. So maybe it does have a big impact on aging. And the best data is that if you ablate senescent cells in the body using a genetic trick in mice or drugs that may have this property, the animals stay healthy and live a little bit longer. Now, so far it's a relatively small effect. It's not bigger than rapamycin for sure, but maybe that's just hasn't been done perfectly yet, um, and the strategies
Yeah.
may not be optimized yet. There's also some concern that the drugs that kill senescent cells or stop the secretory profile may have toxic effects in the body as well. So I think it's still early to determine whether senolytic drugs are going to be a big answer to the aging problem or not. But it's certainly likely that senescence is a contributing factor to aging.
And what about the other hallmark nutrient sensing? Is this
Uh,
related in some way to senescence, the ability of functioning correctly?
well, everything is interrelated. That was the point I made earlier. You can draw a line between any hallmark and any other hallmark, and through some molecular mechanism. And nutrient signaling is a key function in the body, and this comes back to the TOR pathway and other signaling pathways that have been identified that affect aging, and maybe goes even further back to the idea of calorie restriction extending lifespan or intermittent fasting extending lifespan. Um, the suggestion is that your body needs to respond to have the right amount of nutrients in it, and your cells need to respond in the right way to optimize your lifespan. If we think about it from a mouse in a cage perspective, um, generally with those mice, they're getting all the food they want. Whenever they want to eat, the food's right there. This is not how a mouse normally lives its life in the wild. Looking for food is a big challenge for a mouse. And actually, humans are pretty much the same. We throughout almost all of our evolutionary history, our problem wasn't having excess food all over the place. Our problem was getting enough food to eat. And now we're in an environment where we're eating way too many calories. And it's been shown in the animal models, and there's accumulating data in humans, that if you do what's called calorie restriction, which is to bring the nutrient intake down to levels that are just above malnutrition, so you're getting everything you need, but not too much of anything, that it probably will enhance lifespan. Certainly, it does in a mouse or a rat, probably in a human. So, I think
Yeah,
that's a great model, and it's very hard to do though for the average person who's not a nutritionist. Eating a very low caloric intake while still getting the right balance of macronutrients is a bit of a challenge. So people have turned to intermittent fasting. But coming back to your question, um, there are pathways in the cells that respond to these nutrients, and so this is things like rapamycin have overlapping effects with calorie restriction because it turns down this protein called activity of this protein called TOR. Um, and TOR is the protein, or one of them, that senses the amino acids and carbohydrates in the environment and tells the cell what to do. So when you have an overabundance of nutrients, you activate TOR to a very high level, and it tells the cells to rapidly divide and turn down stress response pathways. And when you have low nutrients, it tells the cells to don't divide as much and turn up stress response. And it seems to be that state that's great for healthy longevity. Um, I think that the key thing to remember is all of these things are dynamic pathways. You
exactly.
when you're going through development, you need TOR activity to be high. You're making a lot of cells, you're growing, you're getting bigger. You need TOR signaling. When you become an adult, you're not growing anymore, so you don't need as much TOR signaling. You need it after a meal when there's high nutrients, or if you get a wound on your body locally, you need to activate TOR to repair that wound. But most of the time, you probably need a relatively low state of TOR. So you have a regulation. TORS has a basal state and an activated state. The problem with aging appears to be that the basal state of TOR signaling goes up, so you can't turn it off anymore. So you have sort of rampant signals for nutrient signaling going on all the time. What rapamycin seems to do is to restore that dynamic balance, so you can have the lower basal state, and that's good for longevity. So I think what we're trying to do with nutrient signaling is to preserve the normal regulation that exists in a youthful state that gets lost as we get older.
Yeah,
and
and
and and that seems to be key, and certainly that links to inflammation, it links to cell senescence, it links to pretty much all of the other proteostasis, all of the other hallmarks and pillars.
yeah, this is very interesting transition actually to my next discussion, let's say, point, which is about solutions. So here we talked about the hallmarks, and then you are explaining that there are some suggested strategies in we can apply in our as a lifestyle, like the way we eat, the way we practice. So the promise of, let's say, longevity research as I see it is like high impact on life extension. Let's say sometimes we talk about tens of years, dozens of more years, but when we talk about lifestyle changes, are we talking about that level of impact, or here we are talking more about keeping the same maximum lifespan but just increasing the health span?
Yeah, I think that this is a broader question at the moment with all interventions, and I'll come back to the lifestyle. In animal models, certainly in a worm, we can increase maximum lifespan dramatically, tenfold sometimes. In a mouse, we can increase maximum lifespan 30 or 40%. I think what's unclear is whether we'll be these same interventions, and that could be anything from metformin to rapamycin to lifestyle, senolytics, everything we're looking at right now. Will they increase maximum lifespan in humans, or will they just square the curve? In other words, increase health span and allow more people
Exactly.
to get closer to their maximum lifespan. I think from the standpoint of the population, or from the standpoint of the individual, it doesn't really matter. How much would you pay for an extra 5 years of health or an
Yeah.
extra 10 years of health? If we only extend health span and don't affect maximum lifespan, that's not the worst thing in the world. That's already more beneficial than curing all cancer in people over 50, even if we just get 5 years of health span extension. So this is a massive impact either way. At the moment, I think a lot of the sort of omics-based AI-driven biomarker data may be suggesting that the interventions are not dramatically going to affect maximum lifespan. But that doesn't mean that probably means we're not thinking about it from the right strategy. I do think it's going to be possible to extend human maximum lifespan. I'm not sure whether things like exercise and diet are going to do that, or whether they're just going to square the curve. But either way, I'm trying to do it because I think it's important. If I'm healthy at 100 and dead at 120, that's still a lot better than dead at
Yeah,
80. So it's
of course, that of course makes sense. But the idea is that I think like maybe if good lifestyle strategies really like extend have the potential of extending, let's say 30 years, we would already have seen maybe a human living to that uh,
yeah.
number of years, right?
Um, but it is true that people that exercise live longer than people
Yeah,
that don't, and
yeah,
people that are non-obese and eat healthier live longer. I mean, I think we
yeah,
have some of that data. We haven't seen it optimized. I mean, really trying to optimize everything in one set of individuals would be an interesting experiment. I mean, if you look at the exercise or the diet research that's been done, they don't usually combine the two. And so let me give you an example. I think, there's people are always saying, when you look in the popular press today, eat keto diets, high
yeah,
fat, high protein, no carb. And the animal data all suggest that this is really bad for you. And so the question is, how much amino acids versus carb ratio should you get? Now, if you put a limit on the carbs so that all the animals are getting the same carbohydrates, I'm sorry, put a limit on the calories so all the animals are getting the same calories. Then you adjust the carbohydrates and amino acids. It's the high carb, low amino acid diets where all the animals live longer, the mice and
yeah,
various species. We don't know if that's true in humans. I tend to think that you don't want to dramatically unbalance your macronutrients if you want to live a long time. Now, if you eat no carbohydrates, you'll lose weight, but most people can't stay on those diets, and I'm not sure they're healthy in the long term anyway. But the point I wanted to make is that for amino acids, what is the optimal level of amino acids? Well, I think it probably depends on how much exercise you're doing.
yeah,
If you're doing a lot of resistance training, you're doing a lot of endurance training, you're building muscle mass, that's good. Certainly, high muscle mass is good for a healthy longevity. And you probably require more amino acids to do that. If you're taking very high amino acids and you're sedentary, though, then those amino acids may be driving inflammatory processes and other diseases if they're not utilized to make proteins appropriately. So I think that we need to find strategies to optimize stress, sleep, exercise, and diet all in one go, and then we might see a much bigger effect.
yeah. Again, like we are talking about a complex interaction between the way we eat,
Yeah,
the way we exercise. So to optimize
yeah,
this needs to be really done in a very, let's say, um, studied way, like not
yeah. It's
completely
hard to do though
random.
because people
Yeah,
don't want to make all those changes. The problem with lifestyle,
exactly.
I mean, we've known things to tell people forever, or not for a long time. But it's hard to get compliance because people don't, it's very hard for people to change their lifestyle. And I think that one of the best outlets for lifestyle modification is during childhood.
Yeah,
We have a problem in that these kids, they're more and more looking at their computers. I like computer games too, it's fine, but they're doing too much of that, too much sedentary life. They're stressed at school. They're not sleeping well. They're eating bad diets. This is the formative times for people where if you get them in the right habits, they'll probably keep those right habits. If you
yeah,
get them in the wrong habits, it's really hard to change it back. And so we talk to 50-year-olds that are overweight and say, all right, change all these things. Uh, Most of them won't do it. That's the problem.
yeah. This is true. So here, if we talk about supplement, the current supplements, we usually talk about metformin, rapamycin. You mentioned
Those are
there was,
drugs.
yeah,
I just want
okay,
to differentiate drugs and supplements.
oh,
So, drugs are prescription uh, products that have been developed through research and require a doctor to give them to you. I mean, some people go around that, but you're supposed to have a doctor
yeah,
prescribe them. And supplements, as I define them, would be natural products. So, things
okay.
um,
So about
that your
drugs,
body
so
already
yeah.
produces or that you find in the nature somewhere. So.
Yeah, so let's say metformin, rapamycin, there's also Rejuvent,
Yeah,
AKG.
yeah.
So are those all ways to mitigate the effect of having a bad lifestyle, or let's say a less healthy lifestyle? I mean, if we have a healthy lifestyle, like we exercise, we eat well, do we need those drugs, or they are treating another problem?
That's a fascinating question, and we don't have the answer to that today. Certainly, and there may be differences between different kinds of drugs. I would speculate that metformin works better in people that aren't aging well, at least metabolically. Rapamycin, we don't have a lot of human data yet. It seems to extend maximum lifespan in mice. I think we need to understand that. For AKG, what we found, and this is using Rejuvenant product, which has AKG plus low dose vitamin A if you're a male, and AKG plus low dose vitamin D if you're a female. What we found using, we've done two studies now. One is published, and I can summarize the results without going into detail of the other. If you're We use biologic age as an endpoint, so we can talk about these biomarkers of aging if you want. But what you can look at DNA methylation patterns, so epigenetic changes, and forecast someone's biologic age from that. So if you take their baseline biologic age, give them an intervention for a period of time, and then do a follow up and take their biologic age again, you can determine, at least by that readout, whether the intervention is making
Mhm.
people younger or not. What we found is that people that are biologically at their normal age to start with, or older, so they're not aging well, respond very well to Rejuvenant. If they're already 5 to 10 years biologically younger than their chronologic age, there's very little response. So in that case, it seems to be helping people that are aging normally or not aging well. Of course, that's a huge percentage of the population. But
Sure.
if you're already doing all the right things, or you've got the right genetics, or for whatever reason you're biologically very young, it may have less of an effect. Whether other interventions have the same property or not, I think is unknown at this point.
So,
And it may be
what?
that some things affect some people and some things
Yeah,
not others, and we just don't know why. I think in the end, it's going to be very personalized. We've been spending a lot of time trying to understand the reasons why everybody ages, but now we need to understand why some people age this way and some people age that way, and at this rate or that rate, and that may be differential effects on different hallmarks and pillars, or I think we need to understand personalized aging now. I think that's the next challenge. What's different
yeah,
about me and you?
yeah. The
So
different ways of aging, the
yeah.
mechanism behind these differences. But going back to AKG, so what is this exact? Is it exactly? How is it different from other types of drugs? What are the mechanisms? They are
Yeah,
the
well, we
mechanism of
haven't talked
action.
about NAD yet, but they're very similar. So let me cover both of them at the
Sure.
same time. So AKG and NAD are metabolites. So your body makes tons of this in your cells, and they're used for many different chemical reactions, including production of energy. Um, Both of them go down with age. And so the research from, not mostly not my lab, but other labs, have shown that supplementing NAD, either with nicotinamide riboside or nicotinamide mononucleotide, two precursors to NAD, can improve health span in animals, and there's some human data. AKG, we found that if you supplement with mice starting at 18 months, which is late middle age, you improve not just their lifespan, but predominantly their health span. So what happens is you compress the morbidity of these animals. They stay healthy much longer, and they live a little bit longer. Um, and the human data suggests that it can reverse biologic age in the majority of people. The So they're linchpins in metabolism, essentially, these molecules. If you have less of them, then your body can adapt its metabolic pathways as needed with aging, and they seem to go down with age. I think that another unique property of these molecules is that when you take them, they go up really fast in the bloodstream, and then they go down really fast. It's not like a drug that might persist for 48 hours. These things are used. Your body knows what to do with them. There's hundreds of reactions that they get used up in, and they just get used up quickly and go away. So we think, at least with the AKG, it's important to have a sustained release version where It gets into your gut and slowly releases in your gut over a 12-hour period, as opposed to all just dissolving immediately and getting used up in 20 minutes. That may
Yeah.
be why they're safer too, because natural products are used up so quickly that they don't accumulate in your body like some drugs do. And so the toxicity profiles are very good on NAD and AKG and a lot of other natural products as well, because the body knows what to do with them. It
Yeah.
uses them and gets rid of them.
So this is an approved drug today, or what stage are
So,
we in?
so AKG and NAD, NAD precursor, let's start with AKG. AKG is what's called generally regarded as safe. So that's considered a product that you don't need approval for. You can sell it. Many people sell it, including Rejuven. You don't have to go through all the clinical trials. Now, it depends on what claims you make with a product like that. If you say that it cures diabetes, then you have to do clinical trials to show it. If you say that it's promoting health and over the long term, and you can have data for that to support it, you don't need to. You're not making a disease claim, so the bar is lower on approval. So these products can just be sold over the counter. Think of aspirin,
Yeah.
similar. Um, NAD is also a natural product, but you can't take NAD because it doesn't get through the gut into the bloodstream very well. So people use precursors. Nicotinamide riboside is also widely sold on the market as a natural product, so you can buy that. Nicotinamide mononucleotide, or NMN, is also sold, but the FDA recently limited how it should be sold. They haven't enforced that yet, and that's a complicated discussion. But right now, you can also still buy NMN uh, on online or in stores.
Yeah. So here I'm interested about your work on this subject. So, um, what are your, let's say, points of focus in all the longevity research? So you are working currently on studying drugs, I think. What are the other projects you are personally involved in, and you were personally involved
Well, one of my problems is I'm kind of ADD, and so I'm doing too many different things at the same time. So, but let me stay focused on the main thing, which is to really create, and this is through our Healthy Longevity Translational Research Program at NUS, National University of Singapore, and our Center for Healthy Longevity. We're trying to create a whole pipeline for healthy longevity interventions. The idea is to not just do worm experiments or yeast experiments or mouse experiments, but do things in a way that's coordinated so that we can make basic discoveries, develop translational interventions, and then put them into the clinic and validate that they work for human aging and ultimately get them into the community. So we have about 35 faculty here at NUS working on different aspects of the aging process. I think our lab specifically is really trying to focus on pharmacologic interventions, uh, and really from a relatively agnostic phase. Yes, I like AKG. I hope that works really well. But the main goal is to test many different things,
on?
compare them, and find out what works best. Things that work really well in animal models like mice and killifish, then we move into human clinical studies and try to get validating evidence that they slow aging in humans. In the mice, we're going back now and starting to combine different interventions together, which generates unpredictable results, by the way. I can't forecast which things are going to be additive with each other until we do the experiment. And we also want to start that personalization question in mice so that we let mice age. And then at 1 year or something like that, we start assessing their health and develop specific interventions for each mouse to see if we can do better using that approach than treating all the mice with one intervention. So,
So
so we're trying to push the envelope in the animal models and validate in the humans.
how do you validate that in humans? Like, how do you make sure
Yeah,
that this really gets some effects in humans?
this is a huge question right now, and different groups are taking different approaches. It's kind of at a point where we're not sure what the best strategy is. We just know now's the time to do it. And the reason now's the time, well, there are really two reasons. One is the animal data is compelling. I think everyone believes that we can slow aging in animals. Theoretically, there's no reason we can't do that in humans as well. And so we need to start working on that. the other issue is demographics. The world's aging now. It's not going to 50 years from now. It's not going to matter so much. We're already going to have 2.5 billion people over the age of 65, and we're not going to have enough money to take care of all of them. So we need to solve the problem now, and we need to do that. We need to move into humans. So you have people like Nir Barzilai with the TAME study who are giving people in their 60s metformin and trying to see if it prevents a whole range of different diseases. So that would be kind of a health span trial. Can we prevent many different things with one drug, which is, in theory, what should happen with an aging drug, because aging is driving all these diseases. The challenge with those kinds of preventative studies is they take a long time. So it's a 3-year study. It takes a lot of people. I think the last I heard was 3,000 people. And it costs a lot of money, which, you know, tens of millions of dollars. So this is a drug, metformin, that costs 4 cents a day. It's off patent. Nobody wants to pay for that. And on top of that, the FDA has not yet recognized aging as a disease. So even if you prove you can slow aging, um, then it's very hard to get the drug
Yeah.
approved for that. So I think that approach is great. I'm glad somebody's doing it. I'm very supportive of Nir. But we want to compare many different drugs in humans at the same time, and supplements and lifestyle interventions. And so we're using these biomarkers of age that have been developed recently as our endpoints, and also using metabolic parameters that are linked to the particular intervention. So,
Is
um,
this what sorry? Is this what we call aging clocks?
yeah, aging
Biological clocks.
clocks.
Yeah.
Yeah, and so they can be used from the most well-known ones are based on DNA methylation patterns, so epigenetic changes that happen in your genome. And that can be analyzed from your blood or your saliva or any other tissue, although most people don't want to give up brain tissue for clinical trials. So we're pretty limited to blood and saliva. the, and the You measure the methylation changes in the DNA, and you can predict someone's biologic age. So think about it this way. A lot of people that are in their 50s, right? And
Sure.
so they don't all look the same. Some of them look like they're in their 30s and 40s. Some of them
Exactly.
look like they're in their 70s already. I'm in my 50s. I'm not going to ask you where I look, but the point is that we're not aging at the same rate. So our passport tells us our chronologic age. It doesn't tell us our biologic age. And now these clocks are designed to say, how fast are you really aging? What's your biologic age? So if a true biomarker should predict someone's chronologic age, it should predict the onset of mortality, disease, or mortality in the end. And some of these clocks look like they're doing that. And it's not just DNA methylation. You can do proteomics, the changes in protein levels. You can do microbiome predict clocks now, to changes in your gut bacteria that predict your age. You can look at facial pattern. There's all kinds of different clocks. So what we want to see now is, in a 6 or a 12-month intervention with the drug, we can take someone that was biologically here and move them back to here. And it looks like that's feasible based on the early studies. So those are the kinds of things we're doing. We're using the biomarkers as readouts on age, and not just waiting to see whether people get
Yeah.
sick or not. Of
So,
course, we look at a lot of physiologic parameters as well. We look at things like walking speed. We look at grip strength. We look at VO2 max. We look at so we're trying to combine the clocks with physiologic and molecular parameters that are known already to be linked to aging.
so yeah, talking about clocks, I remember I was in the longevity conference in Berlin, and we talked about machine learning sometimes when we talked about those markers. So
Yeah.
this brings me to the AI subject. So how do you see AI's contribution actually in the longevity sphere? Is it let's say a fundamental contributor? It's a useful tool, or it's not yet clear where AI can help with that
I think
type
it's
of
been
research.
already a fundamental contributor, and really, I have probably 40% of my lab now is doing AI based analysis. And so we're becoming slightly more dry lab every time I walk through the hallways. Now, I may not feel the same way in 10 years when I'm completely obsolete, but for now, I really love AI. The and it's accomplishing many different goals. I think that one of them is that these clocks. So, in 2010, the National Institute of Aging had put a lot of money into trying to find biomarkers of aging. And it basically said it was a failure. We didn't find anything that works that well. Yeah, there are things that predict mortality or age, um, walking speed, things like that are good if you're over 70 years of age, but we need to measure biologic age younger. If you look at individual parameters like CRP, or that's an inflammatory marker, yeah, there's some correlation, but it's not good enough to use really as a biomarker. So basically, what Steve Horvath and many others have done is say, let's take these deep omics data sets or some kind of data set. Let's look at DNA methylation across a wide range of the genome, record all the changes with age, and then let machine learning or neural networking or algorithms predict someone's chronologic age. And then that deviation you get from the prediction is your biologic age. So if the clock says you're 53 when you're really 48, you're not aging well, for instance.
Okay.
Now there's second generation and third generation clocks now that try to predict onset of disease. They are predict your rate of aging, not the current state you're at now. So there's a wide range of options out there, but all of this has come from some kind of AI based strategy. And so none of these are fully validated, but I think they're on the way to being validated, and I think AI has been a big driver in that. It's also being used in other ways, like trying to identify the pathways that are most likely to affect aging that we want to target with drugs, or trying to, you know, understand why some people respond to some intervention and not others. So I think it's going to have a wider range of applications.
Yeah, when we talk about the role of AI, let's say in aging clocks, in the mechanism of action of some drugs or the pathways they affect, how did you use to study those aspects before we knew about AI, let's say? Or these are completely new possibilities that AI opened?
No, I think we wanted to find biomarkers all along, and there would be some evidence that this inflammatory marker is linked to aging. And so we'd measure that marker in 1,000 people and say, yeah, it predicts, age a little bit, but it's a very weak predictor.
Yeah,
Or we would take this metabolic marker, and it was all based on uh, sort of reductionist reasoning based on the literature that was there. And uh, what AI did is kind of what we did with interventions. I think that a lot of us that found the early interventions, we didn't do it. At least our lab didn't do it by saying, "Oh, I know this protein's involved in aging. Let's find a drug that targets this protein." We went back to simple organisms like yeast and worms and just screened every drug we could get our hands on in an unbiased way. And saying, "All right, let's just find the drugs and then work from the drugs back to the pathways." And so that wasn't AI, but it's unbiased. And I think
yeah,
that the unbiased approach of AI is really important. There's a lot we don't know yet. And it's really dangerous to say we fully understand the aging. I mean, even if you look at the reviews, there's different papers. Like there's a new paper on hallmarks that has expanded the list. Some say 20, some say 11. We don't know everything. There's a lot we don't know. We're looking under the
yeah,
lamppost, and what AI does is it um, takes a complex data and it doesn't know which lamppost to look under, so it integrates all of the data and comes up with a potential solution. So I think the challenge of AI is that it says, "All right, you're biologically 56," but it doesn't say why.
yeah,
It's
yeah.
really hard to go
Interpretation.
back to
Yeah,
the mechanism from there, and I think
exactly.
that's what's unsatisfying about it. In the old days when we started working in yeast aging, we were thinking about one pathway or one gene, and we come up with this really elegant hypothesis and a way to test it, and it would be right or wrong. If it was right, we feel like we really accomplished something, trying to
Exactly.
define one little pathway. And now we understand that there's so much interconnectivity between these pathways that maybe that approach is not going to be that useful. but that was satisfying. It felt like you creatively came up with some solution, and
Yeah,
now you're just applying better and better algorithms to try to come up with answers, but you're not sure what questions you're answering. I think sometimes.
exactly, exactly. So interpretation, this is one of the
Yeah,
main problems I personally
yeah,
also work on, how to, if you provide a prediction, then how to interpret that prediction. I'm
yeah,
seeing in my work such a problem in polygenic risk
yeah,
estimation of
yeah,
like the relationship between genes and diseases. So yeah, your point is interesting about the role of AI when we talk about complex data, because many of the techniques see like historical, because we had limited data, limited technologies to, let's say, study linear correlation of some genes with
yeah,
effects. But now with AI, we can really study nonlinear, much more complex interactions, but then the interpretability problems. Very
yeah.
interesting point. Right. Okay. So now let's talk a little bit about progress. So you are mentioning these advances on drug, let's say, discovery in the longevity, and we also know about hallmarks. But I see these, like, let's see these hallmarks like more than 10 years, let's say 20 years from now, we started to talk about hallmarks. We started to develop some drugs, some also techniques to fight against those hallmarks or the effects of those hallmarks. Where are we today? Did we really do progress compared to 20 years from now, or are we blocked? Are there blockers? Where are we actually?
Yeah, I think the big question. I think we'll definitely have interventions that extend health span by then. Um, Hopefully, you lose this video if I'm wrong, but I think we will. And they may have modest effects, 5, 10 years of health span, maybe some effect on lifespan. Um, I feel confident to say we can get there, and already that's a big change. I think the fundamental question is, can we really break the process of aging
Yeah,
and do something dramatic to maximum lifespan? Um, I tend to think that's possible, but it may require a whole different kind of thinking than we're taking right now. Uh, or maybe if we just optimize what we're doing right now and put things together, you'll really get there. But my tendency is to believe that it's going to require some new way of thinking about it. So we're really trying to, in the very basic science aging we're doing, we're really trying to go back to the drawing board and say, are we thinking about this the right way?
yeah,
Maybe we should be finding drugs that have some completely different property than we've been looking at previously. And so I think that's a question that I'd hope that by 20 years we have a good feel for what the answer to that question is, but whether we're actively exploiting that knowledge to make people live to 200,
yeah. This
I
is
think it's unknown still.
yeah. So
But it's great that people are thinking about it. I mean, people often put me in the camp of, I'm just interested in health span, and I'm not interested
yeah,
in living to 200. And I think that I'm definitely interested in that. I think I focused my research on trying to validate that we can have any effect on aging, because I think if we can do that, that'll really increase public awareness and money, and accelerate the research that may solve those bigger problems down the road. But
exactly.
I'm very interested in the thinker, these futurist thinkers and different strategies. And we have our own aging webinar. We have some of those people on our show sometimes just to hear what they're thinking.
Yeah, so here we talk about, say, interventions when we talk about like clinics, innocent cells, or like stem cell therapies. Do you think that there is a potential for those techniques that, let's say, are more intervention, difficult, maybe difficult, more difficult intervention? I honestly have no idea. Can they
Yeah,
increase more
we
lifespan?
don't know. I mean, so stem cell therapy is very interesting. I work with clinics that do that, particularly mesenchymal stem cells. Um, these cells appear. There's a lot of people have done this. They appear to be safe. I think it's feasible that they would affect aging because they persist in the body for a period of time. They're anti-inflammatory. It's not that they're engrafting and replacing your damaged tissues, but they're moving around and reducing inflammation and improving the function of the stem cells, your endogenous stem cells in your tissues. But right now we don't have enough data to know whether it's
Yeah,
affecting biologic age or not. So one of the reasons I want to work with clinics like this is that we can start to accumulate that data to see whether you're really getting effects on health span and lifespan.
yeah. So there was this recent paper by David Sinclair lab about the information theory of aging, and you mentioned
Yeah,
actually the epigenetic
yeah,
aspect of aging. What do you think of that line of work, trying to partial reprogramming or trying to reverse the age cell?
yeah. I think partial reprogramming is very exciting. I mean, that theory goes way back decades, that epigenetic changes are impacting aging and that it's been known, of course, it's obvious that if we get older, but we still make babies that are young. So we know how to keep cells rejuvenated in our germline cells. And so I think that it's not a novel theory. I think that what David's arguing is the impact of this may be huge, and this may be a big driver to solve the aging problem. And I think that it's possible. There a lot of, in fact, when we published the first paper on sirtuins and aging in the 90s, so that's an epigenetic regulator, and there are many other papers that have been published on epigenetic regulators in aging. So certainly they influence the aging process. Whether they have a primacy role or not, I think has to be established. But the idea of reprogramming cells in your body to get back to a more um, stem cell like state, I think is an exciting area of aging research. I think the challenge is going to be how do we do that in a way that we don't go too far and make,
Yeah,
you know, pluripotent cells that turn into tumor cells. And so there's a lot of promise there. How close it is to developing therapeutics, I think, is an unknown. But those are the kinds of fields we need to, or areas that we need to really pursue, because in theory, they may have a much bigger impact than the interventions we're studying today.
yeah. So you said a really interesting comment actually, like we grow older, but we still know how to make young babies.
Mm,
So I honestly, the first time I heard that comment, which is very interesting and obvious,
yeah,
but it makes me ask the question, like, how? Like, I know it's a very basic question, but it's
yeah.
true that we are aging, but we can still get those young cells. Why can we do, like, why can we get this clean DNA, let's say?
Well, our germline cells are preserved and treated in an entirely different way than the somatic cells in our body. And that goes for the DNA and the epigenetic state of the cells, but also from the metabolic properties of the cells. And so they're really in a pristine situation. But because we have to continue as a species or we wouldn't have evolved. If there wasn't a way to do this, we would have all died
Sure.
off. So I think that understanding why that happens is really important. And one of the things we've done here at NUS is start a Center for Reproductive Longevity, where we're really looking at ovarian aging and from several different aspects. What's happening to egg cells? Why do women lose fertility as they get older? Why are ovaries some of the earliest tissues that show signs of aging? And then downstream, what happens after menopause? Why does that accelerate some properties of aging? Why does that make female aging different than male aging? All of these questions, I think, are critical. We haven't spent enough time understanding reproductive aging, and we're trying to go back and correct that now. And also, the idea of doing iPS cells was a revolution. I mean, it got a Nobel Prize very quickly because everybody recognized that taking cells that even old cells from old people, you can reprogram them back to stem cells. Now, there's a big argument as to whether they're really identical to an ES cell or they have how close they are. But certainly you can take those iPS cells that you make and differentiate them back into all the most or all of the other cell types in your body. And so in a way that's replicating that process
Yeah.
in vitro, because you've got primary cells they're going to senesce and stop dividing. But if you put these reprogramming factors, you can make them back to a state where they'll continue dividing.
Yeah.
So that has to be related to aging in some way. And so I think that's why it's so important to really understand that process.
And this is a interesting question about reproductive aging. You mentioned just a bit earlier that sometimes we talk about, let's say, living a few hundred years, but there's always that question: would we be able to reproduce or to make babies all of along that period, or we just will make it for the first, let's say, 50 years for women, especially, and then just go without it for the rest of the time?
Well, that's enough for me. I don't need that many babies. Oh, the I have two, made two, love them, don't need 10.
Yeah,
um,
exactly.
the but I think that's an interesting question. I mean,
Yeah.
I tend to think they'll go together. I think that if we extend lifespan, we'll have longer fertility.
Okay.
If we extend fertility, we may have longer lifespan, and that's one of the questions we're trying to answer.
Okay. So it
Um,
might be solved along the way.
but
Let's
but you're
say.
but you're sort of getting to another question too, which is that what's the cost of extending lifespan and
Yeah.
stopping aging? And and that comes back to evolution. And the Medawar, Williams, Kirkwood, all of these people have thought about evolution and aging over the years. And the take home message is that we're optimized for fitness. We're not optimized to live forever. And that's
Exactly.
probably why it's so easy to find mutations that make us live longer, because it's not what evolution cared about. We're not optimized for that property. And in worms and yeast, 5% of the knockouts live longer. It's shocking. People said you never find a single gene that makes an animal live longer. It's like 1 in 20 in the genome. You knock them
Wow.
out, the animal lives longer. But most of those mutations don't make the animal more fit. They make the animal
Yeah.
less fit. They
Yeah.
they reproduce more slowly. They divide more slow. They grow smaller. Things that would not be good in the wild. And so the challenge really is to find interventions that give you the benefit of not aging, but don't come with some cost. Tendency to get a virus
Mm.
or loss of fertility or other things that affect fitness. And I think there are going to be ways to do that. It's just that we have to think about that as we develop interventions. And in a way, maybe rapamycin is an example of that, because you need very high TOR signaling during development to grow to the right size, to develop in the normal way. But as an adult, you don't need high TOR signaling all the time. And
Yeah.
so if we put a drug in after development, for a gene that's important for development, but is causing
Yes.
problems later, then we might get the benefit without the cost. And it's more complicated for rapamycin and TOR than that, but that's an example of what I'm talking about. So.
This is a very, again, very interesting comment because we optimize for age longevity, but also we need to look at fitness. So bring me to aging clock. When we let's say develop aging clock and test our drugs, let's say, and interventions by optimizing those clocks, but also we need to have fitness markers. Maybe is this something also like cared about explicitly? Let's say in the when we test for drugs, do we have
Well, I
these
mean,
markers?
yes and no. I mean, we look at health span markers, which are one aspect of fitness.
Yeah.
Um, I don't In animal models, we look at fertility. So rapamycin in a mouse will reduce the fertility of a young mouse, but it maintains the fertility longer in a mouse. So it's almost as if, early on it may have a slightly detrimental effect, but these mice are still reproducing later if the
Yeah.
TOR pathway is lower. So it's a trade-off. Um, I think we're not really looking at aging interventions in humans targeting ovarian aging and fertility yet, and that's something we want to start doing. But yeah, I think we need to certainly be aware of what the effects are. Maybe in a perfect environment, if I turn down my neuronal function, I'll save energy and live longer, but I'm not sure we want to do that. So,
Exactly.
it's a
Exactly.
low neuronal function is enough of a problem already. We don't need to get worse. So I think that we need to be cognizant of the interventions we develop so that we don't have unexpected costs that come with the benefits.
Exactly. This is the big question. Okay, so now, um, how people can help, let's say, help themselves or help this research, if it's possible. So people are listening, and they are listening about drugs and uh, trackers. If I'm
Yeah.
listening to that, what can I do actually? Should I take drugs? Is it safe? Should I take many drugs together? Interaction is safe. Trackers, what can we do actually?
So yeah, that's a lot of different questions. So let me answer it at the individual level first. Healthy lifestyle is important. I know it's hard. I know I actually, it's sometimes I think it's harder than people are. It's not as hard as people realize because, like, when I started running, the first 2 months were hard. Increasing the running distance. I didn't feel well. I was tired. Once I got over a threshold and my body was adapted to it, I actually feel more energetic when I'm exercising. And so
Exactly.
I think some of these lifestyle changes have activation energy barriers that you have to get over. And once you're over them, you can see the benefit. And the reason really to do those things is not because you'll be healthy 20 years later, hopefully you will, but it's because you feel better, healthier, more active, more productive now. And so I can't go down the path of drugs without saying that because I think that it's something that people can do. Stress management. How it's not the stress you're put in contact with; it's how you manage it in your brain. Uh, Diet. Get the, don't necessarily do radical diet interventions, but get the calories down to normal, at least, and maybe consider intermittent fasting or eating during time windows of the day. Um, Exercise, resistance training, and endurance training, both are good. Some combination is better, and these things will help you. You'll feel better,
Yeah.
and
So
probably there's some level of change at any point in your life that will be helpful. It doesn't, going out and being a marathon runner at 80, if you've never run before, may not be a good idea.
yeah,
But
yeah.
but a little bit of change can still help.
What about drugs? Is it like
Yeah.
metformin or other drugs you mentioned earlier? Is
So
it safe? How to get them?
so I first of all, I'm not an MD, so I want to qualify that statement. The second one is that um, I'm not advocating that people go get drugs off label that are not prescribed because
Yeah,
I'd like to not be sued. Um, a lot of people are taking metformin. They get their doctor to prescribe it, or they find other ways to get it. This is a No drug is completely safe. There are rare side effects with metformin, but they're pretty rare for most people. It's well tolerated. People are taking rapamycin. That does have side effects, but if it's managed correctly, the side effects can be eliminated or
yeah,
at least minimized. Um, but I'm not sure it's time to go out and start taking drugs right now. I
yeah,
think supplements are a different story. These are safer in general, and there are a lot of people trying NAD precursors, AKG, and other supplements. There's a bunch of them in the GNC store, whatever your supplement store is. Um, none of them are fully validated. Some are better tested than others. Kind of the Wild West. Uh, One thing I would caution, though, for that, I would say if you want to be an early adopter and you educate yourself, and I'm talking about natural products, it's probably okay to go ahead
yeah.
and start trying them. If you want to wait until they're validated in a period of time, that's fine too. I think everybody has to make their own decision. Um,
Yeah, so
I
the takeaway, like the safest way actually, is to live for the moment a healthy lifestyle as much
Yeah, and
as possible,
consider
and
the
wait
maybe
for
consider
research to give it.
and maybe consider the natural products, I think,
Yeah,
but I don't recommend taking
yeah,
prescribed drugs at this point. I would also say that when we do studies in mice putting two things together, I can't predict the outcome. So if you're basing the idea that you're taking these supplements or natural products based on the animal data, which a lot of people are doing because that's most of our data right now, and a single natural product works, but we don't know what happens when you put them together in twos and threes and fours, then maybe you shouldn't be putting them together in
Yeah,
twos
yeah.
and threes and fours
And I
because they
remember
counteract each other sometimes, and we don't know why. So
exactly.
it's that's something we're really actively working on. But I can't. People tell me I'm taking these 12 different things. I have no idea what's going on at that point. So
And
um,
actually, thank you for this comment because I remember watching your talk in the conference in Berlin,
yeah,
and I think you were the only one is to raise that fact that in the lab we are studying this, so
yeah,
maybe it's not complete safe. So it's maybe.
yeah,
So
yeah.
very interesting comment. So yeah, so for the moment, the safest way is to lead a healthy lifestyle, and let's say keep an eye maybe on research and what's getting out from labs and companies.
Yeah, and like I said, a lot of people are taking NAD or AKG or those things are. I think it's okay if you want to try them personally. That's my opinion.
Yeah,
I take the Rejuven, but be cautious. Don't mix a bunch of things together. That's what I recommend.
yeah,
People don't listen to me; they still do it. But I don't know what's happening. I do believe though that people want to inform themselves, and it's really their right to make decisions. It's not my
of
right to tell
course,
them what to do or what not to do. So I'm trying to provide the best possible information out there right now.
exactly, perfect. Okay, so the last question about social impact. I'm not going to talk a lot about overpopulation or these, let's say, classics. I think this is what also I understand from other people, Aubrey Gray, etc., and other people of the field, that overpopulation seems to not be the problem. I don't know if you have a comment on this.
Yeah, I generally agree with that. I mean, there are a lot of people on Earth. That's a problem,
Yeah,
and it uses a lot of resources. I don't think living longer is going to have a huge impact on that because population growth grows linearly if you live longer. If you have a lot of babies, it grows geometrically. And so I think that, the fertility rates have a bigger drive on population than longevity. And even think of it this way, we tend to think of all these potential ethical issues with aging. Even if we could stop aging right now, okay, there would not be 300-year-old people for 185 years.
exactly.
Like, and so, it's not like tomorrow we're going to have a completely different demographic distribution. This is going to, even if you could stop aging, which we can't do right now, that would be a gradual change in demography. So I think that and it's not changing in a vacuum. Who knows what our lives are going to look like 50 years from now anyway. If we're not aging, it may not be that big of an impact given the other technological advances or changes. So, change is not going to be immediate and it's not going to be in a vacuum. So I think that I'm less concerned about that. Certainly, there'll be unpredictable outcomes if we can stop aging or really slow it down dramatically. But I'm one of these people that believes in technological advance. So
Yeah.
it's hopefully it makes us live in a better world. And I don't care what people say. I don't want to go back to the 1950s. So it's
And what about one, like, let's say, one opinion I found interesting is about aging makes people like much more, I mean, many old people living, so it might hinder creativity or openness to new ideas.
yeah.
What?
I'm not sure if older people are less creative. Really,
Yeah,
that's the first comment. Uh,
exactly. So this is actually, to be more precise, comment
yeah,
I read by Elon Musk actually.
yeah,
So I find interesting, not in the sense that older. We know that, let's say, even in the research and like most companies are created by tend to be created by older people. But I mean, let's say about openness, the social, let's say,
yeah,
uh, revolution that happened of younger people, how
yeah.
this type of dynamic that
Well,
might,
so one of the things you also hear this about wisdom. Older people have more
yeah,
wisdom than young
that's
people.
true.
Um, but do these changes come from biologic aging, or do they come from experience?
Very
or
interesting.
or bad
Yeah.
experiences in the case of closed-mindedness. And so I don't think we know. My belief would be that if we slow aging, we're going to slow these changes as well, probably. And you're going to be able to couple people with a lot of experience with very young, active brains at the same time. And to me, that's a positive. If I could take what I know now and the brain energy I had in my 20s and put them together, wow, that would be great. So I think that we're speculating at this point, but I'm not as concerned about it. First of all, I think older people are very creative, and I think that a lot of the closed-mindedness is because society is ageist. It's not necessarily because
Yeah.
they're changes. So ageism is another problem that we absolutely have to deal with. It's not the topic of today's talk, but I think if we deal with ageism, we'll probably go a long way towards solving that sort of social stigmatism that comes.
Very interesting. And yeah, this is true that we have no idea what a 200 years expert brain looks like, what contribution it would be able actually to make to
Yeah,
humanity.
I
So
got in an argument with an ethicist once, and they said, if we live that long, we're going to be bored. And I'm like, well, I'm willing to risk boredom at 150.
yeah, amazing. Okay, so that's it from me. Thank you very much, Brian Kenny, for the
Oh, my pleasure.
fascinating discussion. Thank you for talking on this podcast. And yeah, that's it for me.
It's my pleasure to be on, and anytime you want to talk, just let me know.
Thank you very much.